All right. So again, thank you guys for stopping by today and taking part in our little, I'll just say, scientific discussion on gas chromatography mass spectrometry. As many of you probably know, the science department does have a GC mass spec. It's located in MS 212. And, this workshop presentation is mostly to give you some idea of the things that we have been doing as far as using the GC mass spec, and then hopefully prompt some ideas, promote some ideas for your classes to possibly use the GC mass spec as well. All right. So I know the title's a little longer than what was in the initial description. The actual title was Design and Implementation of Practical Qualitative Experiments Involving Gas Chromatography Mass Spectrometry as an Analytical Tool. And again, if you guys are just joining, please raise your hand during the discussion and you can open mic and ask a question. All right, so to begin, we just want to become familiar first with gas chromatography, mass spectrometry, the instrumentation and operation. I've got a background in mass spectrometry, and so there are a few more details that I'll share. I won't go too far in depth, I hope, but the idea is to let you know what the instrument is capable of doing. Then we want to discuss the types of samples, the sample amount, and sample preparation. That might seem trivial, but this instrument is very sensitive. I can pick up femtomolar, and that's femto meaning the prefix femto, molar amounts of sample. And so it's very critical that you prepare your sample properly when you get ready to use the GC mass spec. And then finally, we got a showcase at the end. We have two projects, one involving CBD oil that we've done with my class, and then one that David will cover. We talk about retention ponds. All right, so let's go ahead and talk about what GC mass spec is. It's an analytical method that combines the features of gas chromatography and mass spectrometry to identify different substances within the test sample. And I point out that it is an analytical method. Of course, everybody knows, I guess, there are 5 areas of chemistry, analytical chemistry being one specific area. I'm an analytical chemist. I know Dr. Carlos Vate Diaz is also an analytical chemist. And in analytical chemistry, we study and use instruments and methods to separate, identify, and quantify matter. And so you might think of this notion of separating and identifying. Well, if you're identifying things, that's the qualitative part. Okay. And so that's what our entire field does. So this instrument is primarily used in analytical chemistry. Of course, it's two things. It's gas chromatography. That's a common type of chromatography used in analytical chemistry for separating and analyzing compounds that can be vaporized without decomposition. As the name states, it's gas chromatography. All right, so you got solid, liquid, and gas. In this case, if the sample cannot be turned into a gas at a regular temperature, meaning up to 120 to 300 degrees, then you probably can't use it in this instrument. And then lastly, here we have mass spectrometry is an analytical technique that's used to detect and measure mass to charge ratio of ions. So here's a little summary of kind of that same thing here. So gas chromatography mass spectrometry is what we call a hyphenated analytical technique, which means it's two techniques that are combined to form one single method. Of course, the GC again is dealing with volatile organic compounds, all right? And the mass spec is our detector. So let's get into a more detail of the instrumentation. This is a picture of the instrument that we have in MS212. There are a number of parts up here that you'll see in another diagram coming up, but I just wanted to point them out so in case you come into the room, you'll know what the system is designed for and have all the parts for it. You've got a carrier gas over here in the corner, so that carrier gas is a helium gas for this particular system. You've got an injection port, and this injection port, I wish I had a video of it, but anytime you watch a CSI program on television, they'll typically have the auto sampler for the GC running because it spins around. It's a little robot arm that picks up the sample and drops it into an area so that you can have a needle pierce it and then inject it into the actual instrument. And then finally, you have this region, the GC itself, then your mass spectrometer, and your data system. So let's dig into these with a little more detail. The first thing we want to talk about is the carrier gas. Okay, so when we talk about chromatography, you got to talk about two different phases. You have what we call a mobile phase, which, as the name is stated, it's a moving phase. And so, in the case of our carrier gas, which sits in our tank, The carrier gas pushes, in our case, helium throughout the entire system. So the carrier gas is hooked up to the injector is hooked up to this gas chromatography column, and then your column goes into your mass spectrometer. So the carrier gases that we usually use are going to be inert gases, meaning that they are not reactive gases. If you put them with other compounds or chemicals, they don't cause a reaction with those other chemicals. So we're talking about things like helium and nitrogen. Now, on occasion, they'll use hydrogen. Sometimes hydrogen's a little bit cheaper, but in our particular case, we don't want to use hydrogen because hydrogen is flammable. So our carrier gas is running through the system. The next area you'll note is the injector port. Okay, so the injector port is that region that I pointed out that was on top of the mass spec in our diagram. It's also got a syringe, and the injector injects the sample into the flow of the carrier gas. And in this region, your carrier gas passes through onto your column. Once the sample gets in, it's immediately volatilized. So it's turned into a gas by a high temperature. Typically, our injection ports are sitting at about 120 degrees Celsius. So if you're familiar with the boiling point of water, you know that water boils at 100 degrees. Most of the samples that we want to inject can be turned into a gas at much lower temperatures. So if you enter that sample, put that sample into this injection port at 120 degrees or 150 degrees in some instances, automatically the sample gets turned into a gas. And so samples can be diluted. We talked about the sensitivity, how little an amount of sample that you have to be able to put into this instrument. We can dilute the sample at the injection port. Okay, so that's one of the details that makes this instrument pretty nice, is that you can do some dilution within the system itself. Next, we're going to go to the GC column, and the column itself is placed in an oven. It literally is an oven. And the oven, of course, can go from 40 degrees all the way up to 350 degrees Celsius. And the idea is that you are slowly heating the column over time. And as you slowly heat the column, samples that are slow to be moving because they're at a larger molecular weight or they have more interaction with the column itself, they actually start to boil and move more quickly through the column. Okay, so the whole separation process takes place as a result of heat and the column itself. The columns are generally about 15 meters to 60 meters long, so that's that coil that you see here in my diagram. And the key there is that the length of the column gives you theoretical plates. That's a principle, but the short end of it is you can think of it as rocks in a river. So the gas chromatography, or the carrier gas, is like the water flowing through the river. And then if you have rocks in a river and you have a small raft, you can easily navigate the river. But if you have a large raft, then it becomes more difficult to navigate all the rocks, and therefore you move more slowly. So certain compounds will interact with the GC column more than other ones. And if they interact more as a result of the column and the compound itself, then they'll come out of the column last. Okay. So columns are a stationary phase that will retain compounds based on how they interact with the column. Just as a side note, when I worked in industry, the guys in industry talked about how they used to take their cans of soup before they had microwaves, and they would sit them in the GC ovens, and they would heat their cans of soup inside the GC ovens. Of course, we wouldn't do that today. There'd be a safety issue with that, but it is just an oven. All right, so the next system there is going to be your mass spectrometer. And, oh, I'm sorry, one more detail about the column. So your things are getting separated on the column. And as they're coming off the column, you can note here, this is a counts versus acquisition time. So the first items that you see down here, about 4.4, 4.6, at about 4.5, this compound came off of the column first. And then you can see successively that each of these other compounds comes off the column at a later time. The idea being that these columns that come off very late, in this case around 7.8 minutes, those columns had a lot of interaction with, or those compounds had a lot of interaction with the column. And so they came off later. And this becomes important because this retention time is one of the things you can use to help identify your sample. All right, so now on to the mass spec. So your mass spec is your detector. All right, so compounds are separated as transferring through the heated area right here between the two instruments. And then you have three parts to the mass spectrometer. You have an ionizer, a mass filter, and a detector. Let me go into that in a little more detail. So over here, you have a GC stream, and your GC compounds, the compounds that are being separated, are going to flow through the entrance of the source. So this is the ionizer, this area right over here. Notice that I've got an arrow here to neutral compounds. So things that come through the GC have no charge, meaning they're not positively charged or negatively charged. They're neutral. When they get into the ionizer, they go through a beam of electrons. And these electrons interact with each one of these compounds and they knock off an electron. And if you remember from your general chemistry, if you remove an electron from a compound, ultimately you create a positive charge because the electron is negatively charged. So by passing through this beam, now each one of these compounds has a positive charge. So that's the ionizer. That's what it does, is create ions. From there, it passes through what we call a mass filter. And there are different types of mass filters, but this one that I have represented here is called a quadrupole. And it's essentially four different rods. And in these four different rods, you will create, or you'll have different voltages. So the top one and the bottom one might be positively charged. The one at the back and the one in closer to the front would be negatively charged. And then you alternate those voltages very quickly. And what that does is if you have something that's positively charged moving through the rods, as they encounter the positive rod, they'll start to move away. But then as they start to go up and you flip the voltage, now that positive charge is on the top rod and they'll be pushed back down. So you essentially filter the positive charges through this region. All right, so that's why it's the mass analyzer. Now we place a DC voltage on this and we scan it, and that DC voltage allows us to get what we call the mass to charge ratio. And basically a mass to charge ratio, if you have a compound like water, and water is a mass of 18, then the charge on it would be 1, so 18 divided by 1 would be 1. So the mass that you would see is 18. All right, so once it gets through there, then it goes on to your detector. And once it gets in the detector, it strikes a plate that causes an electrical signal. All right, and so that's the details of our mass spec. Now that electrical signal goes over to the data system. And our data system has basically two things that it can show. Obviously, you've got your chromatogram that was based on your GC, so you've got all your various compounds that are coming out. And then you've also got a mass spectrum. And in this case, a mass spectrum with your mass to charge ratio gives you different pieces or different ions that are based on the one compound that came through the instrument at a single time. So if we go back to the compound that came out at position or time number 4 on this particular chromatogram, all of this mass spectrum represent that one compound at that one particular time. And we'll get into more detail about that in just a moment. All right. So in summary, you've got a carrier gas, you inject your sample, the gas chromatograph actually separates the mixture, your mass spectrometer detects the compounds, and then your data system collects and analyzes your data. All right. So that's it for the first part. I hope I was good there as far as explaining kind of the components of the GC mass spec. Let me move on to some of the sample preparation. We'll talk about sample state and amount first. And the key there, I said earlier, is that you have to have compounds that are going to be volatile. So compounds must be soluble in a solution for them to be injected onto the GC. The solvent itself has to be volatile, generally organic. So for example, we'll use hexane and we'll use dichloromethane. Hexane, if you guys will note, is a non-polar solvent, while dichloromethane is a polar solvent. All right. The key is here that, however, water being polar, water should not be used as a solvent. Water is a no-no in GC. And that's because if you put water in the instrument, water's boiling point is 100 degrees. And if you were to inject it on the GC column, it would actually crash out as a liquid. And that would be a liquid until it reached the temperature above 100 degrees, at which point it would start to turn into a gas. And then essentially it would fill up the entire column because you have so much of it. Okay, so water we don't want to use. The hexane and the dichloromethane boil at much lower temperatures, and as a result, they'll be off of the column very quickly because they don't need a high temperature for them to go and turn into a gas. For the amount of sample, it's going to depend on the ionization method. In our case, it's electron impact. But the sensitivity of the instrument is about 1 to 100 picograms per component. And so I always tell Dr. Bajet Diaz with his organic class, dilute it once, then dilute it again, and finally dilute it in the instrument because the instrument is really very, very sensitive. The injection amount is actually 1 microliter. And to give you some idea of 1 microliter injection for that liquid, if you inject or push the syringe down, the 1 microliter does not have enough weight to actually drop off the end of the syringe. Okay, that's how small of an amount that 1 microliter is. Okay, so let's move on now to the sample preparation. So sample preparation can change from simply dissolving some of the sample in a suitable solvent to extensive methods, and we'll kind of see that today with the two uh, projects that we'll show. The first thing here with your cleanup procedures, they can take various forms. You can have liquid-liquid extraction. So if you look over here to the side, this might be a simple liquid-liquid extraction. In this case, you might have hexane on top and then water on the bottom. And the water would be drawn out of the bottom of the container. And then the hexane would be drawn out. And then you'd re-add your water. And then your hexane, add a new layer of hexane, and you'd shake this up to actually extract your compounds from your water. That's a liquid-liquid extraction. Or we've also tried solid phase extraction in a couple of places. I know we tried them in geology. In solid phase extraction, you have a solid phase that's located in typically a tube, and you add your sample to the tube, and then using pressures, either negative or positive pressure, you allow the sample to pass through a solid phase. I always tell people it looks like sand, but it's actually silica glass that's been modified. And so you can capture quite a bit of your matrix. The key here is that you want to take as much of the dirty stuff. So, for example, if you literally had dirt, you could mix it up, pour it on top, and then the dirt would stay in the tube, and your soluble components would pass through the uh, the syringe area, and you could collect them. So you want to get rid of the matrix and then put as much of the compound of interest into the instrument. So those are just two methods. There are quite a number, but these are pretty common methods. All right, so how do we perform this qualitative analysis? So first thing here, this is different from quantitative. Okay, I want to point that out early on. If you look at qualitative data just gives you information about the properties or the attributes of your sample. So in this picture, you have different colors, you have a precipitant. This can tell you that a compound is there, but it does not tell you quantitative data, which is how much of a sample you have. So maybe in the future, we'll give a presentation about doing actual quantitative analysis with the GC mass spec. But when we're talking about qualitative, we generally just want to figure out what compounds are there. So qualitative analysis, your chromatography, this is all from your data system. Chromatography gives the retention time for your compound. So in this particular case, this is a Chem 151 sample. They are asked to make aspirin from salicylic acid. And in this case, the student did not do so well. The salicylic acid can be found here at 8.35. And you can look at the mass spectrum. This fragmentation pattern that you have over here tells you that you have salicylic acid. All right, so those are the two main ways that you'll identify the compound. The first is the retention time, and then the other one can be the mass spec pattern. So for your GC, if you have a compound, for example, the salicylic acid, you can't really identify the compound without the mass spec. And so what you'll do is you'll inject a standard. And so I would go to the lab and I would get a standard of salicylic acid, and I would inject it using the same chromatography conditions, and it would give me a retention time. And then when I inject a sample of unknown, if I have a compound that comes out at the same retention time, then I can probably say that that same compound is the standard or is the same as the standard. In this case, it would be salicylic acid. Okay. So as long as the GC method is unchanged, compounds will elute from the GC column at the same or very similar retention times. So that's why we use our standard. As far as the mass spec is concerned, we can use a database. So we'll go over to what we call the webbook.gov.nist. That's the website. And you can type in salicylic acid. And when you type that in, it'll give you this page. And right down here, it'll say the mass spectrum for it. And so you can look at that mass spectrum and match it to your mass spectrum. And GC fragmentation is very specific. It creates the same fragmentation pattern for that compound each time. And so we can match this fragmentation pattern to the fragmentation pattern in the database, and that will tell us that we have that compound. So here's that example. This one's from the NIST webbook. So you see here the 138, 120, 92 for the salicylic acid. In our GC sample, you see the 138, the 120, and the 92. And the abundances, meaning the intensities, are about the same. So that tells you that you probably have the salicylic acid, right? If you do not have a database, then you have to interpret the mass spectra or have a known standard of the compound. If you want, we can do some discussion later about how to interpret mass spectra, but I can tell you that's an entire course within itself, all right? So let's move on now to the qualitative analysis. We've got two different projects that we've been working on. The first one here is qualitative analysis of cannabinoid, which is also known as CBD. And then I'll let David introduce his a little bit later. That's the qualitative analysis of VOCs in retention pond water. So first thing about CBD, I think most people have heard of CBD. If you go to the grocery store, I know in the checkout lines at the grocery store, they have CBD products. I'm pretty sure they're sold in the malls. I'm pretty sure they're sold at the vitamin shop, the GNC. You can order them from all over the place. So it's become a very hot topic. GCMS analysis can be used to look at the components found in any of these CBD samples. And chemical structure is one of the objectives for general chemistry. And so I have over here to the side two different CBDs. Now, if you look up CBD, you don't find these two structures. This is actually from the NIST website. And for those who are savvy, they notice that everything else in these two structures is the same, except for this little wedge that's on the top. Here, this wedge is solid, and down here on the lower one, it's a dashed wedge. So if you took Dr. Vajadiaz's organic class, you would know that the wedge that's solid means that this front part is coming towards you, and that's a chiral center. And on this lower one, the wedge or the dashed line means that this part is going away from you, and that's another chiral center. Okay, so it looks like there are two different CBD components. So in this case, we'll look at this in a second, but we want to pay attention to that. Please remember that you have two different CBDs there. So the sample preparation is just dilution. In this case, you have a chromatogram of our CBD samples. We have a standard that came off the column at about 14.54 minutes. And so this is a known standard. We just diluted it and put it directly in the system to check and make sure that we had retention time and that it was what we say it is, the actual CBD. Results for the CBD compound show a few other components, right? So this particular sample on the bottom is a sample that we ordered, and this sample has more peaks in it than the standard, which it should, right? Because CBD is a natural product. So the additional peaks that were found in the sample, we tried to identify those using mass spec. And what we find is that the peak that's at the back is THC. Now, I point that out because I'll show you here in a moment that the FDA has a level. Cannabinoid is not, or I'm sorry, the cannabis plant, the hemp plant is not a marijuana plant. And so therefore it has a level of THC in it, but you should not have a huge amount of THC in it. You also point out that these other two peaks, this is the peak at 14.54 that is the CBD standard, but I found another peak at 13.96 that's also a CBD. So if you look this up in the database, it has a very similar mass spectrum to the other mass spectrum for the CBD. So there's the instrument itself seems to have resolved two different CBDs, but there's only one that we ordered for the standard. Okay, so without the mass spec, we wouldn't know what these other components are. Okay, and just as a side note, just because we're dealing with CBD here, I'm not going to read all this to you guys, but I do want to point out that CBD is a single component found in the cannabis plant. And because it is, these other components are probably there. So when students get in my classroom and they start to talk these things, I point out to them that there are a ton of other things, kind of like the tea tree oil had a bunch of stuff in it. There can be a number of other things that are found in these things. The FDA has only approved one CBD product, and that's to treat things like seizures, these other type of things up here. I know the seizures are a big deal. You can actually go out and Google CBD PBS, and they have quite a number of videos that talk about how CBD is used to treat seizures. And then there are a couple of other details down here, but the key is that CBD itself is a product where it's one compound, but a lot of the things that students are talking about have a number of other things that can be shown with GC mass spec. All right, so let me switch over. David, did you want to fill us in on your background for your retention pond water?
Yeah, so I had this idea a while back because in my Geology 111 class in lab, I do a flood hazards lab. And if you weren't aware, these ponds that we have on campus and all around Eastern North Carolina are a very simple way to uh, control flooding. So what they do is they store the storm water temporarily, and they let the water soak into the ground more naturally versus storm drains that pipe that directly to the rivers, and that can cause flash flooding. So I had this idea that since we're draining this impervious surface, aka parking lots, where we can have vehicles that leak all sorts of petroleum, that that possibly over time that these stormwater retention ponds could be point sources for pollution. It means we know exactly where that pollution came from. So we've got some results of that we if we spike the sample we can retrieve it, but we have not yet been able to detect it in the water. Now you can see on the upper right hand corner of the screen there, you can see this is right outside the where I park every day, and this was right after a rain. So that is oil that is on the ground, and so you can see that that is going to eventually end up into the retention pond. Depending on the results of the water, I would also like to do some coring of soil around the ponds to see if we can detect any petroleum in the soil as well. But because of laboratory contamination, we're having some issues with getting some good results. Hopefully, at some point in time, we'll get this instrument called a purge and trap, where it does the extraction for you and injects it into the GC mass spec, so the points of contamination is dramatically reduced. But as of yet, we've not been able to acquire that piece of equipment, and that's all.
All right, thank you, David. And like you said, I mean, we've got a project or a method for this, and that method is very involved. That's kind of why we want to try and get the purge and trapped. The method itself is a liquid-liquid extraction, and this is just a diagram of liquid-liquid extraction. I kind of talked about it earlier. But if you've been to the chemistry lab in room MS212 and even in the prep room in between, you've probably seen these huge 2 liter separatory funnels. And what we do there is we'll add in about 500 milliliters of pond water or retention pond water, and then we'll add in maybe 30 to 100 milliliters of dichloromethane or hexane. And you shake your sample with those particular solvents. And then, of course, the dichloromethane is more dense, so it would be on the bottom. Hexane would be on the top. But either way, you let the eluents out of your separatory funnel, and then you might do 2 or 3 other extractions to try and get your sample cleaned up. And the key here is that you want to concentrate your sample. Because the pond water is going to have such a small amount of compound in it, it's important to take almost a liter of sample and concentrate it down to maybe a milliliter. So you're doing 1,000-fold concentration there. These are some of the details of what we're using. So we use a deuterated standard for analysis. And when you hear the word deuterated, that means you have 3 different isotopes for hydrogen. Isotopes meaning that you have more neutrons. It's the same element, but you have more neutrons in the nucleus. And a neutron is going to make it weigh a little bit more. It's going to make it weigh 1 atomic mass unit more. And so we have a deuterated standard of octane, because of course we'd be looking for things that are gasoline type, so octane itself. And we pass it through that liquid-liquid extraction procedure. The key is that we can spike that octane standard directly into our water sample before we start to do the extraction process. All right. So results here are just from the mass spec, your standard, and then the mass spectra from that particular compound. So here's the compound itself. And what you want to think about is for every single hydrogen that you would have on the octane, you're going to get 1 deuterium. And so that deuterium is going to add 1 additional mass unit. So what that means is that you can put this particular compound into the extraction, and even if octane is in the extraction or the pond water itself, then you can still have them separated by mass to charge. The mass spec can tell the difference. The GC cannot tell the difference, but the mass spec can tell the difference. The retention times for the 2 compounds would be the same, but the mass spec tells you it's different. And the big thing there is that later on we'll use this standard for qualitative or quantitative analysis because we can take peak areas and we can normalize things because we know how much of this particular compound that we are adding to the sample. And so we can use that to figure out how much we've recovered when we take it through the extraction process. All right, so that's our pond water analysis. These are other applications up here that are just from literature, and hopefully you'll see one up there for you. Let me encourage everybody to, if you want to use the GC mass spec for any of your laboratory work, please let me know. There's probably a technique out there that we can use, even if it's plants. I know I've worked with Mr. Horn in the past on trying to do things for tomatoes. We've also had a number of other things, like I say, the aspirin lab. If you want to do kinetics, we can do things like that. So with that, I think that concludes everything for us. Does anyone have any questions? Please. It's quiet as a mouse. Okay. All right. Well, if you guys have no questions.
Connell, I got a question.
Yes, sir.
If could you use this technique to measure ethanol concentrations like produced by fermentation?
Yes, sir, you can. In fact, that would be a great project because that's just a specific GC. There's an actual GC method for looking at the various alcohols.
Mm-hmm.
And so we could just switch over to that method, make the injection, and go from there. But yes, that can be done very easily.
All right, thanks.
Other questions? I see Carlos has one up here. You can also use an IR instrument to do that. Yes, he's right. You can also use the IR instrument to do that. And we also have the LC instrument that you might be able to do that with as well. So there are a number of analytical pieces of equipment, Mark, that we might maybe can use to do that analysis. Any other questions
Connell,
for Mina? Yes, ma'am.
it's Armina. I had a question. You spoke of how GC fragmentation is very specific.
Mhm.
However, and I'm trying to word my question as best I can based upon my understanding of your very um, very good presentation. Are there any redundancies among chemicals?
So are you saying are there fragmentation patterns that are similar?
Yes,
Yes,
among
and
compounds.
yes.
Because,
In fact,
I'm sorry, even
go
with
ahead.
the example you presented, um, yeah, I think you interpreted my question correctly.
Yes, yeah. So yeah, the compounds themselves, the two that I presented have chiral centers. So
Hmm.
those were resolved because we had the GC in front of them. However, without the GC, the mass spec could not tell the difference. And so that's the key is that the mass spectrum for each of those compounds would look the same, but we know that they're different because they had different elution times on the GC. So you are correct that you'll have very similar patterns. And I see Dr. Carlos actually pointed out that hydrocarbons have very similar patterns. They do. Hydrocarbons break apart based on the number of carbons that they have. So, for example, if you had octane, you can expect to see between 8 and probably 6 groups that are separated by 14 Daltons in the mass spec. The issue becomes that there are different arrangements of those hydrocarbons. So it doesn't have to be a long chain. It can actually have side chains and things like that. Generally, side chains will break off and you'll have a very significant gap when you actually interpret the mass spectrum. But you are correct that you can have some, and we use those groups, the similarities in the mass spectrum to say that yes, you do have a six-membered ring, for example, you have a benzene ring fragment that's very well known. Or if you break off a hydroxyl, which means you have a loss of 18 because you have hydrogen and oxygen, and then you add one hydrogen to it, so you can lose 18. That specific loss tells you that you have a certain pattern. And so again, it would takes a little bit to be able to interpret mass spectra. In fact, there's a lot of books on how to interpret mass spectra, but they do, in some cases, have a lot of overlap.
Okay, great, thank you.
Mhm. And I see Carlos's point up here. You know, that's a fascinating thing, Carlos. I would like to get some understanding of what's going on in that GC mass spectrum. It was identified, Carlos is saying that those enantiomers, the two different CBDs, have very similar structures. So theoretically, we were not doing chiral chromatography. So the only thing that would separate them would be their boiling point or their interaction with the column. And so that's a real question that I have is, you know, was that first peak, it showed up as CBD? I need another standard of a different chiral center CBD to say for sure that it is a different CBD. That's a good point. Yep, the diastereomer would have a different boiling point. That's correct. So those are the details in there.
That, and I thank you, Dr. Valle, for your interjection because I think that's where my thought was going when I asked about the redundancies. I just didn't know to relate it to the enantiomers or the diastereomers. So thank you.
Yep, good science. Are there other questions? Any discussion points? Anyone to add? Anybody want to add anything? No. Thanks, Carlos. All right. Well, if no one has anything else, let me encourage you guys. You know, if you think of anything, like Mark was saying, look at fermentation, or even if you want to look at constituents of some plant that's in the greenhouse or anything like that, or if you're doing, I know we're not supposed to use human blood, or if something like that comes up, we can try and always run them on the mass spec and think of some different projects to be used on the mass spec. So please let me know if you have any ideas, and I'll be around. Thank you guys for coming by today. I appreciate it.